So let's continue again. So Mendel also took yellow colored seeds of pea plants, crossed them with green, and what he discovered that in this monohybrid cross, yellow was dominant, green was recessive, but otherwise the relationship between yellow and green was again the same. So in F2 population, he had 3:1 ratio between yellow and green. Okay. Again, you know, 2 impure yellow, 1
excuse me,
pure
sir.
yellow, 1 pure green.
Slide share नहीं हो रही.
Sir,
Slide
screen
change नहीं हो रही?
sharing.
No.
Do you see the slides now?
No.
No, sir.
No, sir. You
No,
have to.
sir, the slides नजर नहीं आ रही.
Just a moment. Do you see my slides now?
Yes, sir.
Yes, sir.
Do you see slide changing now?
Yes, sir. Yes,
Okay.
sir.
So what I was telling you, in his cross between yellow and green, he discovered that yellow is dominant, green is recessive. Just like cross between round and wrinkle, the ratio in F2 was also 3:1. The underlying genotypic ratio of F2 phenotypic ratio was again 1:2:1. So it held true in all 7 different traits he observed in the pea plants. What he did next was he said, what happens if I take 2 traits and follow them across different generations? And what he did, he decided to take yellow, green, okay, and round, wrinkle traits, and he selected 2 parents. And what he did, he set up a cross between yellow, wrinkled, with green, round. In the F1, he was expecting I should get yellow, round, and that's what he got. Then he self-crossed the yellow round, and what he discovered a ratio which was F2 ratio was what we call 9:3:1 ratio. And 9:3:1 ratio means he received yellow rounds, yellow wrinkled, green round, and green wrinkled. If I put numbers, I mean, from where these ratios actually were driven, after self-crossing yellow round, the numbers he obtained round yellow. These numbers were 315, round green were 188, wrinkled yellow were 101 and wrinkled green was 32. When he decided to follow two different traits, and such a cross is called dihybrid, when you are chasing two different traits, dihybrid cross, involving complete dominance. The question he asked was, while two different factors are being passed on to the progeny, and then from the first generation goes to the second generation, are they impacting each other's inheritance? Are they interfering with each other or supporting each other? Do they have a preference that if I write down the genotypes, so yellow was this one, wrinkled was this one, green is this one, and round was this one. Is there a preference that, when we have F1 will be, you all should know, this one will be F1, yellow round. The round is dominating recessive allele, the yellow allele is dominating green allele. When you set up self-crossing, this one, the question he asked whether there is any preference that yellow will be passed on together with just this one, or this one with this one. And when he saw these numbers, he said, let's consider them as a monohybrid cross. For a moment, we don't look at yellow or green. We just add up all the rounds. And rounds were 315 plus 188, and wrinkled were 101 plus 32. Then he forgot about the seed shape, round and wrinkled, and he said, let's look at the colors. And yellows were 315 plus 101, and green were 188 plus 32. If you add them together, you will get these numbers. And what he discovered, that I may be wrong here with this number, but what he discovered that the underlying ratio between round, wrinkled, or yellow green, even though in the F2, the parents of F2 were capital Y, capital R, small r, the ratio remained intact. Round, wrinkled, yellow, green. So there is no change in the underlying 3:1 ratio, which was originally received in the monohybrid cross. Which means underneath these 9:3:1 ratios, there are two hidden 3:1 ratios. And what they are highlighting, if I write down now here, yellow, wrinkled, green, rounds, what will be the gametes here?
Sir,
My
capital
request
Y,
to all
small
the
r.
biologists is, all the ones who have taken biology, they remain quiet. And someone who has never taken biology, please speak up and dictate me the gametes. What will be
Sir,
gametes?
एक तो कैपिटल Y हो जाएगा और एक R, ये पूरा गैमीट हो जाएगा.
ठीक है.
And then Y R एक दूसरा गैमीट.
इसी का होगा?
नहीं सर, इसका एक और बनाना होगा ना, बिकॉज मियोसिस में दो हमारे डिवाइड होते हैं.
या वेरी गुड. तो कितने गैमीट्स बनेंगे इसके?
नहीं नहीं YRR 2 times हमारा रिपीट होगा ना बिकॉज वो डिवाइड हो रहा है
ये भी आ गया, और कितने होंगे?
जी
बस
बस that's it I
ठीक है।
think. And then you just do the same thing for the other gamete as well. So Y capital R, that's one gamete. Y capital R, that's another
ठीक है,
gamete.
सो हमारे पास F1 आ गया, कैपिटल Y ये फ्यूज हुए, स्मॉल y R। अब इसको सेल्फ किया तो इसके कितने गैमीट्स बनेंगे?
capital R, small y.
उन्हीं को जवाब देने दें, बिकॉज वी आर गोइंग टू लर्न बाय डूइंग। सेम स्टूडेंट हु वाज आंसरिंग पेरेंट्स के गैमीट्स, सेम स्टूडेंट प्लीज आंसर F1 के गैमीट्स। आपका नाम क्या था?
जी सर इर्तजा
इर्तजा। जी इर्तजा।
जी so I'm sorry but could you possibly repeat your question? मेरा नेट जरा स्टटर कर रहा था तो I couldn't hear कि
या
आप
सो
क्या
व्हाट
कह
विल
रहे थे
बी गैमीट्स ऑफ दिस पेरेंट F1 जब हमने इसको सेल्फ क्रॉसिंग की है?
इसको सेल्फ पॉलिनेट करके किया है
हाँ जी, ये एक पेरेंट है ना, तो इसके
ओके
गैमीट्स क्या बनेंगे?
Y एक गैमीट बन जाएगा आई गेस
Y बन जाएगा,
और Y and then R small r एक और गैमीट बन जाएगा
और
if I'm understanding this correctly and then that's it ये तो हमारे तो बन गए then you do the same thing for the other parent as well so capital Y small y and capital R small r
so in meiosis when we start from a diploid cell, how many products of finally कितने इससे बनते हैं cells meiosis के बाद?
Two.
we meiosis we go from a haploid diploid to haploid now
How many haploids we get after complete
so we
meiosis?
get two haploids basically
Okay. Did you go and revise meiosis? I asked all of you.
Yes, sir.
So, who
Yes,
else?
sir.
And someone who hasn't taken biology and is taking first time biology, or let's say has taken biology in O levels but did not take biology in A levels, he or she please help me solve this. G. Khozema.
Sir,
Yes, sir. I think there will be 4 haploid cells produced from one diploid.
Have you
First, the
taken biology
diploid,
before?
I took it in O levels before.
Good. So there will be 4 gametes, 4 haploids from a single diploid. That is what the product of meiosis is. So when we made gametes here, we have to have 4 gametes, although they all are going to look same. And what is the other condition, Khuzema, in meiosis? Haploids. So which means if we had 46 chromosomes here, each of them will carry 23.
Yes.
There is another condition. Look at this one. Look at this one and answer.
Each parent has a 50% chance of having a contribution to the gamete.
Now, think deep. Is this gamete okay for you? This one, the one I'm encircling, or this one?
No, the homologs will be separated. So capital Y, capital R will be a single gamete, not capital Y, small y.
G. Iman, some Iman Atik. Please.
yes, I just wanted to comment on. I'm not sure whether these 4 gametes are. I'm confused at how can they be correct since both the genes and they're for the same trait. For instance, the capital R and the R is for the seed shape. So shouldn't it have one factor of each? So a gamete should be something like a capital Y and a capital R and a capital Y and a small r. and then a capital R and small y, and then
Yeah, that's what good that you are also think. So that's what Josema. So the student who was talking to me earlier, was he Josema? Am I right?
Yes, sir. I was.
So Iman Josema pointed out that two homologs cannot be in the same gamete. So one thing we should know that the chromosome numbers from diploid are half to 23, and we get 4 gametes from one single diploid. The second important concept we should know about meiosis is that the two homolog chromosomes, they go to opposite poles. They will never be on the same pole. So I try to draw it here. If this is carrying Y mitotic chromosome and its homolog is small y, the first thing in meiosis 1 happens is the homologs, they move to opposite poles. And what we have in meiosis 1, I'm again and again repeating meiosis 1, because then there's meiosis 2. In meiosis 1, the homologs will move to opposite poles, these metaphase chromosomes. Then this cell undergoes meiosis 2, and these sister chromatids in meiosis 2, they undergo simple mitosis kind of thing, where the two sister chromatids are separated, and what we have a haploid with YY, a haploid with capital Y, capital Y. So the two homologs will never go together like previously what we had, the gamete, the previous student who helped us, he said we'll have a gamete, capital Y, capital R, small r. I intentionally wanted to solve this with you so that you learn by doing mistakes. So unless we don't do mistake, we are never going to learn things in life. So here, as we are learning, the two homologs are never going to go in the same gamete, which means capital Y can go with R, capital Y with small r. Similarly, small y with capital R, small y with small r. Same gametes with, because it's self-pollination. If we now draw a checkerboard, write down the gametes, now I write down the genotype and you speak up the, all of you write down the, speak up the phenotype. The phenotype?
Yellow round.
Good. So keep speaking as I'm writing the genotypes. यार बोलते जाओ.
Yellow round.
सारे yellow round हैं.
सारे yellow round हैं. Yellow
Yellow
wrinkled आ गया.
and wrinkled. Yellow wrinkled.
Yellow round. Yellow wrinkled.
Yellow round.
Yellow round.
Round.
Yellow round. Green
It's
round.
still
Green round.
green round.
Green round.
Green
Green
round.
round.
Yellow round.
Yellow round again.
Yellow wrinkled.
Yellow
wrinkled.
Green
Wrinkled.
round. Green wrinkled.
Green round. Green round.
So you remember we got 9:3:1 ratio, which was from these numbers, okay? And here, when you look at these, you will get 9 round yellow, 3 round round yellow, 3 round green, 3 wrinkled green, and then yellow green. wrinkled green. This is the homozygous recessive parent 1. Green, wrinkled. So out of this, what Mendel concluded that, and I try to draw it here, according to our latest understanding, that if I have two different traits, yellow and round, so the shapes of chromosomes are different, so that these are different chromosomes. If this is F1, this is the cell when it will undergo meiosis. In meiosis, what happens? During the DNA replication, we learned about the DNA replication. We get complete duplication of the genome. Sorry, capital Y, not small y. We get complete duplication of the genome. And this is how at metaphase chromosomes look like. These are homolog chromosomes. Position of gene on a specific position is called locus. Position of Y on chromosome is called locus. The position of round, the locus is like this, and this is wrinkled. Now, in meiosis 1, what happened? We have capital Y, okay? And it can be sorry, not here, here. Like it can be either this way, or it can also be capital Y, small r. Or in meiosis 1, these are possibilities, okay? Or it can be small y with capital R, or the possibility is small y with small r. Whatever the possibility is, let's stick to one possibility. Let's say it went like this. Now, this one is now undergoing meiosis 2. And in meiosis 2, what we are going to have? We are going to have capital Y together with capital R. No, why I wrote it like this. The second one will be its sister chromatid to a other daughter cell like this. And if this was the possibility where now we had the small y, because we have to decide capital Y went with capital R, its other product will be this one, the homolog of capital Y carrying chromosome went with small r, okay? And the gametes will be now small y r, small y r, okay? But when we look at these numbers, what we are concluding out of these crosses, that capital Y, this one, it can go with capital R, capital Y can go with small r, small y with capital R, and small y with small r. In a population, the end result will be this one, 9:3:3:1 ratio. The conclusion which we draw from this experiment is that when we are following two different traits, the two different traits controlled by two independent genes which show complete dominance and recessiveness in between two alleles, during inheritance from parents to offspring, that's what the Mendel's conclusion was, that they assort completely independently. What it means, assort independently. This is independent assortment. They are, shuffling. One thing is clear, capital Y and small y are never going to go to the same gamete. If they end up, there will be a big disaster in the progeny, in the offsprings. One thing is they are going to go to opposite pole. This is what his law of segregation was all about. But when we have two different genes controlling two different traits, now they assort independently like this or this or this. G Zainab.
Sir, he said that both of like capital Y and small y can't go in the same gamete. What's the exact reason for that? Like, ये नहीं होगा कि if it does go, तो एक वाला would be dominant and the other one would just not play any role. What's the disaster specifically that will happen?
Good question. Very good question. So let's learn it like this. We are like this, okay? And Zainab said, what will happen if we end up having capital Y, and both of them going together? R are separated. This one carries only R because homologs, they are reduced to half in meiosis 1. But Zainab said, let's think about if capital Y and small y homologs, they ended up in the same gamete in meiosis 1. Now, let's just focus on capital Y. So how it looks like? Capital Y, small y, small y. This is how this in metaphase will look like. Now, when this will undergo meiosis 2, the resultant gamete will have capital Y, small y. The other daughter gamete will have capital Y, small y. Now, we know these are gametes, haploids. They are to be fertilized. Let's say the oocyte, let's say these are sperm cells, or oocyte, vice versa. The oocyte, the egg cell, has capital Y and let's forget about R because it will confuse you. Now this gamete, when it will fuse, because this is haploid, this is not haploid for this particular chromosome. Now when fusion of gamete will take place, the zygote will look like this. Capital Y, oocyte has also capital Y, and there is a small y here as well. This is disaster because you have 3 copies of same chromosome. Each chromosome in a diploid exists in pairs, like they have homologs. There are 2 copies of each chromosome. Now, what you will have, P has 7 chromosomes, which is haploid number. Diploid number is 14. In this organism, in this zygote, how many chromosomes, Zainab, you will have?
Sir, which one?
This one.
So it will have 3, right? So 21. You said 3
No.
copies of the same, right? So
3 copies of the chromosome which carries Y allele, not every chromosome. Round is on a totally different. So there are 7 chromosomes. Chromosome 1, the 2 looks like this. Let's say the 3 chromosome looks like this. 4 chromosome looks like this. 5th one, and then 7 chromosomes. Let's say this one carries Y, the other R may be present here. There are, I don't know, n number of genes in thousands in P, which are distributed in all these chromosomes. We have only 3 copies of this one. All the others, they got half. They got reduced to half in meiosis 1. So we have 3 copies of only this chromosome in this zygote. How many total chromosomes we will have in this diploid now?
very bad at math. Let me think.
30.
You asked a very good question.
So will we have 38? No, wait, we'll just have one more chromosome. So one more from the diploid configuration. So isn't that just 47?
47 कहाँ पे हो गए? ये P में मैं कह रहा हूँ 14 क्रोमोसोम होते हैं डिप्लॉइड में. We started diploid 14 थे यहाँ पे. तो यहाँ पे 14 होने चाहिए ना बेसिकली तो नेचुरली. लेकिन यहाँ पे आप कितने होंगे अगर Y और Y होमोलॉग सेम क्रोमोसोम में चले गए?
एक उसका जो है एक एक्स्ट्रा कॉपी बन रही है तो सिर्फ वो वाला मल्टीप्लाइड बाय 2 होगा, राइट? बाकी सारे तो वो हाफ ही रहेंगे.
लेकिन डिप्लॉइड में 7 इधर से आए, 7 इधर से आए, बल्कि हैप्लॉइड में 7 इधर से आए, इधर से कितने आए? 8. 1 copy of each and 2 copies of this one. 1 2 3 4 5 6 and 2 of this 8 हो गए. मायोसिस में 14 का हाफ जो ट्रू मायोसिस नॉर्मल होगी उसमें 7 होंगे. तो 7 + 8 कितने हो गए क्रोमोसोम? 15.
So basically
Now-
one more than the standard diploid configuration. Now ideally हमारे 14 होने चाहिए थे, but अब इसकी वजह से 15 हो गए.
Exactly. Now, why this is disaster? Have you heard about Down syndrome?
Down syndrome इससे हो जाता
Yes, sir.
है. Basically
Extra
Down
chromosome.
syndrome is we
Chromosome
call
at
chromosome
23rd number.
21.
आपका एक extra
तो जो जैनब ने पूछा है, जैनब अगर वो सेपरेट ना हो तो ये डिजास्टर होता है. क्रोमोसोम 21 की 3 कॉपीज हैं और डाउन सिंड्रोम वाले बेबीज में 47 क्रोमोसोम होते हैं. हम लोगों में 46 होते हैं. सिर्फ एक कॉपी एक्स्ट्रा होती है. डू यू अंडरस्टैंड व्हाई द डिजास्टर वाज?
Yes, sir, I do. Thank you
Okay.
so much.
So,
Sir?
yeah,
Sir, I had a small question. I was confused about the
we have a quiz
like
now. So जिसने मैसेज करना है कि यार आ जाओ चीज वंडी दी लभन लगी जे ते मैसेज कर दो दोस्तां नु ताकि आ जानो सारे.
sir,
हाँ
can
जी
I ask?
बताएं. Sorry.
I just had a small question. I was confused that, sir, like when we say homolog chromosomes, do they like code for one sort of like trait, like say for round or wrinkled, or like does the same chromosome actually have like like can it code for different traits as well? Like what does homolog basically mean
Yeah,
in this
homolog
sense?
means identical chromosome, contains same DNA code, but structurally, physically, they are 2 independent structures.
Yes,
We are
sir.
just
Then I was
considering-
basically
Let-
like
hold
yeah,
on.
like this here.
We are considering that on one of the homolog there is green allele, on the one other homolog it's capital Y allele. For simplicity, we are ignoring everything else, but on these chromosomes, we will have other genes too. In humans, consider this as human genome. Let's stick to the P genome. We are just for simplicity and to explain you, we are just considering one trait or one gene here. But there will be
Sir?
hundreds and thousands of genes on one single chromosome. On our 46 chromosomes, which are 23 pairs, 22 homologs and one is X or Y, or in females 2X chromosomes. There are nearly 20,000 genes. Now you can imagine how many genes each chromosome may have. Did I answer your question?
No, sir, I'm just confused about the fact because, like, say we have capital Y and small y, they obviously would have different protein sequence, right? Then, like, what does it mean to be homolog if they're like, even their sequence is basically different?
Very good. These are not homologs. These are alleles of same gene. Clear? This is homolog. Chromosome, the word homolog is being used for chromosome. Homologous chromosome. When we say homologous chromosome, we are referring to two identical, nearly 99.9% identical pieces of DNA, which contain same set of genes.
So, like, so we can think of it like this: that both the chromosomes would have the same place for, say, the round or wrinkled gene.
Exactly. So if yellow is there, it will be exactly same position of green here, because these are same genes. They will be on the same position on the chromosome. But these other horizontal lines, these are depicting or representing other genes, which we are ignoring for our analysis. Clear?
Okay. Yes, sir, that makes sense.
So let's have a quiz now. What time is now? Yeah.
Sir, it's interesting discussion, chale. Let's just delay the quiz.
Oh, don't worry, my dear.
Yes, sir, इस तरह की discussion को delay नहीं करते हैं.
Don't worry, we'll have this discussion in next two lectures.
So we shouldn't interrupt this discussion.
Oh, I love your comments, gentlemen. We'll have this, don't worry. So let's go to your LMS, log into your LMS, and we will continue with this discussion, because this discussion is going to enter into what we call chromosomal theory of inheritance. So Mendel's work was moved forward by T.H. Morgan, which is Abdullah's favorite. He named our fly lab Morgan's lab, Morgan room. So Morgan questioned, what happens if I have yellow and wrinkled, green and round on the same chromosome? Because it was Morgan who proposed, or before Morgan, people proposed that Mendel had no idea what we are talking about. Mendel did not know genes reside on chromosomes, this thing, homologs. Mendel just postulated factors. And Mendel was simply a genius, okay? But later on, 40 years later, they discovered that gene factors, which Mendel discovered, they actually reside on chromosomes. Now, the question Morgan asked was, what if the two different genes, they reside on the same chromosome? Will we still get 9331? Okay.
Sir,
So let's
sir,
go to
उसमें तो
LMS,
possible है.
and we will continue this interesting discussion in the next lecture. Okay, जी सुमेर.
सर वो जो मेंडल ने इनिशियली करना था कि उसने वूल बेहतर क्वालिटी बनाई थी, वो फिर उसने बना ली थी.
देखो, जो साइंस है ना, साइंस इज अ लीनियर जर्नी. वो वूल इतनी जल्दी नहीं इंप्रूव हो जाती, लेकिन 40 इयर्स लेटर दे डिस्कवर्ड कि अच्छा ये फैक्टर्स हैं. और जब आप किसी कोई प्रोडक्ट बनाने की कोशिश करते हो, वूल इंप्रूव करनी है, तो वो याद रखिएगा कि बेटा वो इटसेल्फ कोई 50 साल का प्रोजेक्ट होता है. इतना आसान नहीं होता. यू हैव टू आइडेंटिफाई जीन्स व्हिच आर गोइंग टू एनकोड वूल. पहले तो वो आइडेंटिफाई करोगे ना. वो आइडेंटिफाई करके फिर आप अच्छी क्वालिटी वाला जो जीन है उसे क्रॉसेस के थ्रू लेके आओगे. सो आई होप यू हैव लॉग्ड इनटू योर LMS. एंड अब्दुल्ला, आर यू देयर?
Yes, sir. So I have just published the quiz. Go to LMS, वहीं पर जहाँ पे सारे quizzes होते हैं.
and you will have 3 minutes to do 2 questions, and it will be linear.
It's
That's
a
it.
very
Yeah, it
simple
will be-
quiz. Just 2
It
questions.
will be available in the next 30 seconds.
Very simple. Trust me. Allah Hafiz.
Sir, I still can't hear it though.
Yeah, it will be there at 4:30 na. 30 seconds hai abhi.
4:40.
It's a very simple quiz based on what we have learned in the last 2 lectures, 3 lectures. Don't worry. And each quiz is hardly, I believe, 0.5 or 0.25 marks. I don't know exactly. But don't worry. This is very simple quiz. Allah Hafiz.
Allah Hafiz, sir.
Allah Hafiz, sir.